Hey everyone, and welcome. Today we're diving into a huge topic in the scientific community, the reproducibility crisis. I mean, think about it. How can we trust research if we can't get the same results twice? It's a real problem. But the good news is we're going to look at a super practical, step-by-step solution called ARTE, which is all about making your work transparent, solid, and, you guessed it, truly reproducible.
So, here's what we're going to cover. First, we'll dig into what this reproducibility crisis is all about. Then, we'll introduce the RTA workflow as the solution and break it down into 3 different levels of rigor. We'll also unpack what the gold standard actually means and wrap up by talking about why this is really about building a new culture, not just learning to code.
All right, so why is this such a big deal? Well, this whole crisis has really shaken people's confidence in science. When other researchers can't verify your findings, it kind of undermines the entire foundation of how we build knowledge together. And this isn't just some abstract academic thing. It has real-world effects on everything from public policy to new medical treatments.
Let me ask you this. Have you ever tried to rerun an analysis from a paper you read? You download the files and it's a total mess, files are missing, the code makes no sense, and you just can't get the same results? If that sounds familiar, you are so not alone. It's a massive issue, and it usually comes down to disorganized projects, missing software versions, and just a general lack of transparency.
And to really get a sense of the scale here, just look at this number. A survey in the journal Nature, a pretty big deal, found that a whopping 70% of researchers have tried and failed to reproduce another scientist's experiment. 70%! That's not just a small glitch in the system, it's a systemic problem that we absolutely have to fix.
Okay, so we've talked enough about the problem. Let's get to the solution, and that's where ARTE comes into the picture. Now, this isn't just another complicated piece of software you have to learn. It's a structured workflow, a template designed to bring some much-needed order and clarity to your research right from the very beginning.
So what is ARTE? It stands for Article Reproducibility Template and Environment. Basically, it's a pre-built project structure that uses common tools like Quarto and RStudio. The whole point is to give you a clear, logical system to follow from the get-go, so you aren't stuck reinventing the wheel and trying to figure out how to organize your files every single time you start a new project. ARTE is built on this really simple but really powerful standard called the Tier 4.0 protocol. It instantly organizes your project with 3 main folders. You get a data folder, which smartly separates your raw, untouched data from the cleaned-up data you actually use for analysis. This is so critical because it means you never accidentally overwrite your original data. Then there's the scripts folder for all your code. And finally, the output folder, which is where all your results, like figures and tables, get generated by your code.
And this right here, this is the magic concept at the heart of it all. The idea is that your output folder should be completely disposable. You should be able to literally drag it to the trash, hit delete, and then just run your code from the scripts folder and have every single figure and table pop back up perfectly recreated. If you can do that, you've achieved true computational reproducibility. That's the test.
Now, here's what I think is the best part. ARTE isn't some rigid, all-or-nothing system. It's designed to meet you wherever you are in your journey. It offers this really flexible path with 3 different levels of rigor, so you can start simple and then build up your skills as you go. You can think of these levels kind of like a spectrum. You start out at minimum, which is all about good organization. Then you can move on to adequate, which adds more control. And finally, you can aim for complete reproducibility. Each step just adds another layer of strength to your research, making it that much more transparent and easy for others to verify. Level 1 is all about just getting organized. Seriously, there are no complicated command line tools here. You just use the ARTE template, sort your files into those data, scripts, and output folders, write a little bit of documentation in some simple text files, and then share the whole project on a platform like the Open Science Framework. It's the absolute perfect first step, and anyone can do it. Okay, at level 2, we start adding in some more powerful tools for version and package control. We bring in Git and GitHub, which basically creates a full time machine for your project, tracking every single change. We use a tool called Quarto to link the text of your article directly to your code, which means no more copy-pasting figures and introducing errors. And this is huge. We use a tool called REnv to record the exact versions of all your R packages. This completely solves that classic, well, it worked on my machine problem.
And then we have level 3. This is the gold standard. Here, we use a technology called Docker to package up your entire computing environment. I'm not just talking about your code and data. I mean everything. The operating system, the specific version of R you used, all of it. This creates a completely self-contained capsule that is guaranteed to run the exact same way on any computer anywhere in the world, anytime.
So
what does that really mean to have complete reproducibility? It sounds kind of intimidating, right? Let's take a closer look at what's going on in level 3 and demystify this whole idea, because the concept behind it is actually pretty simple.
Honestly, the best way to wrap your head around Docker is to think of it as a digital time capsule for your analysis. It literally freezes your entire research environment, the software, the code, the operating system, into a single, neat package. So 10 years from now, another researcher can come along, open up this time capsule, and get the exact same result that you did, without having to worry about broken dependencies or outdated software. It's amazing. So here's the key difference between Level 2 and Level 3. At Level 2, Renv is like a checklist for the books in your library. It makes sure you have the right R packages. But at Level 3, Docker manages the entire library. The building itself, the shelves, the lighting, the operating system. It ensures your analysis works on any machine, period. No excuses.
As we start to wrap this up, it's really important to zoom out for a second. We've talked a lot about some cool tools like Docker and Git, but the real power of a system like ARTE isn't in any single piece of technology. It's about fundamentally changing how we approach our research from the very start.
This quote really just says it all. The specific tools we use, they're going to change over time, but the principles, organization, clear documentation, transparency, those are timeless. By adopting a workflow like ARTE, you're helping to build those habits, and that creates a culture of reproducibility. That is the real, lasting value.
Now, if you're feeling a little overwhelmed by the idea of Docker, don't be. The creators actually recommend a really practical approach. It's called end use for encapsulation. Basically, you just do all your work normally at Level 2, and then you only use Docker at the very end to package it all up and make sure it works. It's a great way to dip your toes in the water. The most important thing is just start somewhere.
So as you start thinking about your next research project, the question isn't whether you're ready to jump straight to the gold standard. The real question is much simpler. Are you ready to start at Level 1? Just by using a template to organize your files and sharing your work openly, you are already taking a giant leap forward toward a more open, robust, and trustworthy science.